Hello everyone, my name is Sahan. I'm the admin of Helix Weekly Digest website. Today I decided to carry out an experiment called Rna Extraction on Proxy Labs. Proxy Labs is a virtual platform that gives you the opportunity to perform the labs related to chemistry, physics, and biology. I'm gonna focus on the experiments related to biology, of course, starting with Rna Extraction. I'm gonna do the rest of the experiments for every weekend later on. RNA extraction is necessary for real-time PCR with the COVID-19 in the corner. I'm sure most of you have heard of this name. It's also used in northern blotting, macroarray analysis, and forensics and other fields. In this experiment, we isolate and separate RNA from DNA using Trizol. Trizol is an acidic solution containing guanidinium thiocyanate, phenol, and chloroform. I'm going to talk about the function and purpose of using phenol and chloroform during the experiment in the relevant section. But bear in mind that guanidinium thiocyanate is a general protein denaturant. It denatures the DNA. How it denatures the DNA, I'm gonna need to go back to the molecular biology. As you learned in the molecular biology, protein has four different types of structure: the primary structure, secondary structure, tertiary structure, and quaternary structure. The primary one is the basic one. It's a linear sequence of amino acids linked together by peptide bond. It written mostly in three letters or one. It starts from the N terminal of the first amino acid, goes toward the C terminal of the last amino acid. The information regarding this could be obtained from gene sequencing or encoding protein or by protein itself. This is more detailed picture, including the peptide bonds. This is the C terminal of the first amino acid. This is the N terminal of the first amino acid, and this is the C terminal of the last amino acid. The bond between carbon and oxygen here is peptide bond, as well as the bond between the nitrogen and hydrogen. As you remember from the chemistry, or let's say from the periodic table, oxygen is in group 8, it's non-metal, it wants to gain electron. Uh, and hydrogen is in group 1, alkali metal, it wants to lose electron. So these two are likely to make another bond called hydrogen bond. When such thing happens, there is gonna be two kind of different protein folding. The first one is gonna be shaped like a helix. It's called alpha helix, and the second one is gonna be like a zigzag, like this. It's gonna be called beta sheet. Bear in mind that this is how the secondary structures are of the proteins are. So to go back to the first picture, tertiary structure is same as the secondary structure, but differences there are more other types of bonds participating in this, such as ionic bond, disulfuric bind, hydrophobic interactions. If you remember the cell membrane structure, there are fatty acid tails that they hate water. They are hydrophobic. They're not permeable for water or charged molecules such as ions and stuff. So this is type of interaction we mean. The quaternary structure is quite self-explanatory. When one tertiary structure is interacting with other tertiary structure, they form new proteins by using dimer, trimer, and tetramer. Without further ado, let's carry out with our experiment. Firstly, we're gonna use 70% of ethanol to clean our working area. We're gonna spray it around. Then we're gonna dry it and scrub it over. Then we're gonna need to use 3% of hydrogen peroxide atomizer. Same as before, we're gonna spray it around. We dry it, are cleaned up, and ready to carry out our experiment. What we're gonna do, we're gonna need to add 1 microliter of Trizol. We take the micropipette, we add our tip, then we take the Trizol agent, we fill it to the tube, we get rid of the tip of the pipette, put it back. We put the cap back on. Then the next step is to homogenize this. We're gonna mix it thoroughly. Now that it is mixed, we're gonna leave it for 5 minutes to incubate itself at the room temperature. Then we need to take the content of the tube and throw the tube away and add a new one. The new one is added. We fill it back, we get rid of the tip, we go that way. Now at this part we're gonna add chloroform. We open the cap, we're gonna need 200 microliter of it, I think. Yes, that's correct. We take the tip, we take the chloroform. I'm gonna talk about the reason why we're using chloroform shortly. I want for you to get a better picture so that I could explain better. So we added that, we put the cap on, we inverted twice to mix it. Then we're gonna centrifuge it. We add another tube filled with water to maintain the balance. We're gonna centrifuge it for 15 minutes in 4 degrees Celsius for 12,000 RPM. Here's the timer. That's done. I'm gonna take a moment and talk about what happened till now. We added Trizol reagent. If you remember, I talked about the guanidinium thiocyanate. That's the denaturant. And what it does, that it precipitates DNA along with RNases and other proteins. So they're gonna be in the lower layer, and RNA is gonna be in the upper one, aqueous layer, which is colorless part. Later on, we added chloroform. Chloroform. The purpose of adding chloroform was to remove the fuzziness between these two layers. Then all we care here is about the RNA one, which is the upper layer. We're gonna take our micropipette and we're gonna take that part. We're gonna be careful not to take from the lower part. That's it. We're not gonna throw the red part away. We're gonna store it at 4 degrees Celsius to use it to isolate DNA and our protein. So new tube added. We can feel it. So we're gonna need to take the tip away. Then we're gonna need 500 microliter of isopropanol. The reason that we're adding isopropanol is simple. We want the isopropanol to precipitate with RNA. How it happens is simple. Actually, it forms hydrogen bonds with water and replaces that between RNA and water, thus participating it. We close the cap. We throw the tip away. We're gonna mix it 2 times. We're gonna leave it for 10 minutes at the room temperature, then we're gonna centrifuge it to get all the DNA in the bottom layer. At the end of centrifuge, you will see what's happening. With the same setting, it's gonna be for 15 minutes, 4 degrees Celsius and 12,000 RPM. As you can see, by the end of centrifugation, we will able to see the total RNA precipitate as white pellet at the bottom of the tube. That's the part we really care. So we're gonna take the supernatant part away. We don't need it. We're gonna throw it away. We're gonna add 1 microliter of ethanol to wash it and remove any salt or phenol that is remaining in the white pellet. We added that. We close the cap. We're gonna mix it 2 times. Invert it. That's done. Then we're gonna centrifuge it again to get most of the RNA that we can. We want a pure RNA, but this time with different setting. It's gonna be for 5 minutes at 4 degrees Celsius, 7,500 RPM. That's it. Now we're gonna take the supernatant part again. Gonna throw it away. We're gonna use the now we're gonna dissolve the RNA in RNase-free water. You're gonna need 50 microliter of that. It's gonna be different pipette. Take it. Then we take our RNase-free water. Then we add it. We're gonna mix it. Happen. We close the cap of that. We get rid of the tip. Then we're gonna give it a water bath. It's gonna be for 60 degree. 60 degrees Celsius for 15 minutes. Well, this way we have concluded our experiment by extracting RNA and dissolving it in RNase-free water. We're gonna store it at minus 20 degrees Celsius for upcoming videos. In some of them we're gonna use it, and in some not. We're gonna need proteins or the DNA, which we also put it at minus 4 degrees Celsius. So we're done. I hope you guys enjoyed it, and stay tuned for the other videos. Thanks for watching. Have a nice day.